Thank you. Okay, so thank you, Heather, for bringing me here. So my job today is to give you tips and give a conceptual framework for growing with, for working with algal cultures. And when I was asked to give this talk, I thought, well, what does the ocean acidification community need to know about, algal culturing? So I'm going to try and start from scratch and go back to what phytoplankton need and what we are giving phytoplankton in the cultures, what kind of things we need to look at when we are growing this organism. So here you have some cultures of phytoplankton, typically clonal cultures that we use to test our hypothesis. These clonal cultures, more often than not, we assume that they behave like the way we describe the species concept for that particular functional group. For example, I work on Emiliania haxillii, for which you can see a bloom here. But these blooms are composed of many different genotypes or physiotypes, ecotypes, phenotypes, whatever you want to call them. And what I'm trying to say is that the behavior, the morphology, the physiology of these different types can be very different. So I'm just going to try and cover that. And if you have any questions, just stop me through the lecture. So this is what I'm going to cover. So I'm going to start with just talking about phytoplankton and what we need to know about them, the basics in culturing algae before you get started. What do you need to know? How do you need to plan your experiments? I'm going to talk about knowing your organism, how important it is to understand the physiology, the behavior, if you like, of your mother organism. I work on phytoplankton and on sea urchin larvae, so I need to know pretty well how the physiology is controlled by the environment, right? So I'm just going to talk a little bit about this. The method of manipulation, just very briefly, because people have been discussing this in depth here, other speakers. Some sampling considerations, especially with regards to the new omics, if you like, technology. So for example, I work on proteomics. Some people are working on transcriptomics and genomics. So the way you need to think about your sampling in order for you to be able to interpret the data later on, because the samples, the sampling for some methodologies requires you to work really fast and in very specific conditions. So I'm going to go through this, and then of course some evolutionary considerations that are going to be a central theme through my talk. So for those of you who don't work on phytoplankton and are thinking about it, we have eukaryotic and prokaryotic species of phytoplankton or groups of phytoplankton. So the eukaryotic species have got a defined nucleus, so they are more complex, and then we have the bacteria, some photosynthetic bacteria that are key primary producers in the oceans. They live in the upper layer of the water column. The diversity, the structure, the abundance, these populations are controlled by nutrients and light predominantly, and of course they are transported in the water column as well. So one has to take all these things into account when studying natural populations and when deciding which clones or which populations to target for isolation of what we call clones or strains of varieties within a population. Some species form blooms, but other species are non-bloom forming. And we tend to study the bloom forming species, but I will give you an example of how some non-bloom forming populations are actually more important biogeochemically. So if we care about fluxes, the direction and magnitude of fluxes, perhaps we should think about the species or the strains that we target for conducting these kinds of populations. And then another consideration is that on short time scales, phytoplankton growth and division are tightly coupled with the circadian cycle. So we have to keep this in mind for the way, in terms of the way we do sampling, when during the diel cycle we sample and how many times we have to sample during the diel cycle. And I'm going to briefly mention, and this is my personal view on the parameters that we need to sample as well as the traditional ones. This is just a representation of the of productivity. So this is pigment concentration in milligrams per cubic meter. So you can see that there are these hot spots, and some of these populations at specific times of the year are largely monospecific. So you get one bug forming blooms. We assume that they divide asexually, vegetatively, but actually there is a lot of there is supporting evidence that shows quite clearly that blooms are generally not clonal and that there may be more sexual reproduction within the blooms and therefore more diversity. And then we tend to look at individuals representing what we call phytoplankton functional types or functional groups, and we tend to label them or cluster them based on their size and their structure within the population. So more or less diverse bloom and non-bloom forming species, blooms or populations containing hundreds of coexisting species, or populations containing just one single species, like in the case of Emiliania huxleyi, which is my favorite bug, and this is my model organism. But I also work on other groups like the dinoflagellates, and one is to take the evolution of these organisms into account because they are incredibly complex, and that complexity is required for you to understand changes in the growth of these populations. So here is a bloom of a red tide, and I'm going to show you why they are complex in a couple of slides. So what do they need? Well, they need light to grow, so these are the main factors controlling primary production. They need nutrients, and in terms of nutrients, what we do in the cultures is we supplement seawater, or we make artificial seawater from scratch, and we add a source of nitrogen, a source of phosphorus, trace metals, and some vitamins. You may have axinic and non-axinic cultures. I do not believe in axinic cultures in my condition, so I just don't grow axinic cultures, but I keep bacterial levels as low as I can by subculturing them very often and by maintaining very diluted cultures. So my cell densities are extremely low when I maintain my cultures in the culture collection that we have at the NOC. So I do not use antibiotics, but of course it depends on the questions that you are asking. I think it's extremely hard if you use antibiotics sometimes to interpret the results, and I just stay away from that. So if you want to work on axinic cultures, there are techniques that you can use, but you have to have an extremely well-controlled lab, and we only have one chamber where we can do that. So it depends. For the purpose of my questions, I do not need axinic cultures. I can work well with very low levels of bacteria. So temperature is important in selecting for species. So different species have different thresholds of tolerance to temperature and light and so on. So temperature will determine the presence of specific of particular groups or species, and then the physical processes will disperse the species in the water column or the populations. So these are the main processes that you will find in the natural environment. But in terms of the evolution of these organisms and why we care, for the questions that we are all concerned here with, how organisms adapt and how organisms acclimate to environmental selection pressure, I think this is quite important when you work in a vessel, when you have a culture of phytoplankton. So here you have diatoms, and these are the three eukaryotic phytoplankton functional types in the modern oceans. So diatoms, coccolithophores, and dinoflagellates. And they evolved through time over Earth history to give these groups. This happened because of endosymbiotic events, and we cluster them as functional types. So it's kind of like biogeochemical guilds rather than based on their phylogenetic or taxonomic affiliation. So we call this a silicifier, and we call this a calcifier, and these are dinoflagellates with specific functions, like some of them produce toxins, some of them are bioluminescent, but these guys are really interesting in that they have genomes which are up to 100 times larger than the human genome. So they evolved to give these huge genomes, and they can switch. They are kind of like in the twilight between the animal and the plant kingdom. So they can switch from being photosynthesizers to being heterotrophs. So they are incredibly interesting, but they are a pain in the neck when you grow them in cultures, because if the conditions are not, if the conditions switch to being stressful, some of them form cysts, for example, So this is one of the reasons why whenever you grow any of these organisms, you really have to look under the microscope. And very often we just forget to look under the microscope. I work on proteomics, so I just do my technique and harvest the material and do what I have to do. And you still have to look at the, at the cells, because sometimes you can get contamination, you can get a change in the life cycle, and that is going to affect your signal. So you need to know about these things. So this, although this is not central to growing cultures, we need to keep this in mind, because of the the complexity of these organisms is great. Another thing I wanted to mention here that I nearly forgot is that, so these are diatoms, these are silicifiers, but some of these actually fix nitrogen as well, So we also have to keep this in mind. These are calcifiers, but when they form gametes, when they undergo gametogenesis and when they form the sexual cells, the cells do not carry any calcite. Yeah, so even if you don't care about the life cycle of the organism, you need to look at which stage of the life cycle they are going through, because the vegetative cells are carrying calcium carbonate, and the gametes substitute the calcium carbonate for organic scales. So this information is really central to the questions we are discussing here. So knowing that, and of course, another thing that I want to say is that, I'm a biologist, and I always have the species concept as central to everything I do. But the species concept for what we are working with is sometimes not that useful, because there are a lot of functional differences between between organisms within what we call the same species. Yeah? So we have to just be critical about it, be aware of that, and just use that information not based on the name of the organism, but based on what they do. So in some ways, I think, Richard Dawkins' selfish gene, thinking is very appropriate to us, because, he thinks about organisms as mere gene carriers, as carriers of functions. And this is the way I try to work with these organisms. So, if they have a function, then, they are a high calcifying strain of EHAX or a low calcifying strain of EHAX or whatever. And that is much more useful than just working on the species concept. So one of the main problems that we have in the kind of work I do is that when we run incubation experiments, I run them in 2 weeks or in a month at the most, right? And you do the pre-conditioning of the medium, the pre-acclimation of your cells, and then you just run your experiment for maybe 10 generations or something like that. But we are giving these brutal exposures to changes that would take place over maybe a century, and you are working on generation times that are equivalent to maybe, I don't know, a month in the oceans or a year, let's say. So we are working with these cultures, giving pulses that are not realistic in terms of the rates the rates of change that these organisms would experience in the natural environment. So this is something that I am thinking about to design my experiments. If I want to test adaptation or acclimation, long-term adaptation or long-term acclimation. So if you are working with, coccolithophores, for example, or even diatoms, and of course bacteria, they have generation times. Let's say they divide once a day. Some of my species divide 1.5 times a day, some others 0.6 times a day, and so on. But on average, let's say once a day. And if you work with bacteria, they divide every few hours. So you have a beautiful model system to be able to test, if you like, evolution in a bottle, kind of thing. So you can plan this experiment. So this is very much what I'm working on, in my proposal to NERC. In the evenings and very early in the mornings now. And we are trying to figure out a way of testing a century of evolution over 1,000 generations within a vessel, and do de novo sequencing every 200 generations and so on. So these are things that we can begin to think about. Of course, we need to go to the field and make observations in the field, but these kinds of experiments are extremely useful because you can take advantage of these generation times. And evolution is defined based on mutation rates, based on rates of evolution. So you can theoretically plan these kinds of experiments accompanying observations in the field. Of course, this is in contrast with other organisms like foraminifera and theropods, and I'm talking here about calcifiers. So with these organisms, we have two problems. One is that they divide less often, that they reproduce less often than autotrophic organisms. and then it is very hard to maintain them in cultures. Yeah. So the beauty of working with these autotrophic model organisms is that it's relatively easy to work with them, and they divide very rapidly. Okay, so so the basics in culture in algae, I'm going to look at the screen better. You have to decide on the type of cultures that you are going to use. So you can use batch cultures, which I will show you in a second. You can use semi-continuous culturing approaches, or chemostats, or continuous cultures. Yeah. So depending on your question, you might want to choose one or another. Of course, for these long-term evolution experiments, you definitely need continuous cultures. You need to decide on the variables to monitor, and this is stating the obvious, but the best thing is that you plan your experiment well in advance, you consult your colleagues, and you measure as many variables with as much replication as you possibly can. You need to ask yourself whether you have sufficient information about your model organism for the experiment that you are doing. Yeah? So when you make assumptions about the way the organisms use carbon, for example, and I will give an example later, you need to be sure that if you are giving a manipulation, you need to know whether you know or you don't know the answer, and then you design your experiment accordingly. For example, that was my question yesterday, I think, with regards to calcium. How much do we know about the upper and the lower threshold for tolerance levels of these organisms to calcium, and like calcium with many other things. So you need to be aware of these things. You need to think about your control. What is your control in your test? Another thing that you might want to think about is whether you can compare your data with published data. Of course, sometimes published data have behind lab work which uses methodology which is obsolete or that you may want to alter, but sometimes it's good to think about the conditions other people used in past experiments for you to be able to compare your results with. And of course, before you get started, it's good to talk to your colleagues, and I would say colleagues from different disciplines. I'm a biologist and I more or less know about what I need to test, with my cultures, the variables that are important. But if you talk to geologists or chemists, sometimes it's really interesting to talk to them and get information about additional things that you can look for. Okay, so we have batch cultures, and this is one of the main approaches that has been used in phycological research over the last century, I would say. So all these are all the stages of growth. So you have the lag phase, the exponential phase, the stationary phase, and the senescence phase. Yeah. So they undergo these different phases based on the mainly in nutrient conditions. So then most of the experiments that you read about are conducted in exponential phase, just because it's easy. You have this fast grow, exponentially growing culture, healthy cells, sometimes not so healthy cells depending on the conditions, but most of the experiments are conducted here in this phase. Yeah. So for example, if you are doing an experiment in which you want to look at whether organisms are a source or a sink of CO2, coccolithophores, for example, most of the experiments deal with this part of the curve. I haven't come across any experiment that does a budget based on the entire growth curve, on the entire population development. So these are things to bear in mind. And in these experiments, the conditions at time zero are very different from the conditions at the end of the experiment. Yeah? So the cells run out of nutrients. Also, the cells grow to levels which are quite unrealistic, because in the natural environment, you have much lower nutrient concentrations. Yeah? So just to give an example, most studies until, I don't know, a decade ago have used F/2 medium, which has 880 micromolar nitrate, which is, it's about an order of magnitude at least higher than what the organisms could experience in the natural environment. So, you have cell densities which go beyond, way beyond what you would encounter in the field. So there is some self-shading in the media. So you have light limitation and nutrient limitation and so on. So these are not ideal to run for any form of experimentation, in my view. So then you have the semi-continuous cultures, and in these cultures you have the lag phase, just like in the batch cultures, followed by the exponential phase. And what you do is you maintain the cultures exponentially growing. And of course, you establish the growth curve before, so you know the division rates and how long you can maintain them exponentially growing. And then you subculture them halfway through the exponential phase. And then you have the cultures always here, always exponentially growing. And the conditions at time 0 are different from the conditions at the end of the experiment, or at time n, if you like. So this would be time n. But the rate of change is constant, so you always keep them there exponentially growing between a constant, with a constant range of cell density. So this is what I do in the lab. It's a lot of work because you have to subculture them very often. And if your cells grow fast, you have to subculture them even more often. And these are continuous cultures. And in the continuous cultures, you have a range of cell density and a range of nutrients and a range of all these parameters that vary within a narrow range. And the conditions at time 0 are similar to the conditions at the end of the experiment. So you keep them at a constant rate of growth and rate of, ideally, calcium carbonate formation and organic carbon, photosynthetic carbon fixation, and everything. Or nitrogen fixation if you work with nitrogen fixers and so on. So you keep them, at a constant rate and with a very narrow range of cell densities. For this, you need to have chemostats, and you need to have sensors in your chemostat, and this is ideal to run long-term incubation experiments. So, what we have been doing in terms of testing the effect of climate-relevant variables on phytoplankton physiology to look at the potential effects on biogeochemistry, we use lab cultures, we use mesocosm experiments, and we do DEC experiments. So the lab experiments have been at the center of the phycological research over the last century, and then more recent work used exactly the same approach, just varying the conditions and varying some of the techniques, but basically doing the same thing. Then mesocosm experiments have also been done. They are seen as halfway between the lab and the field. I'm only going to talk about lab cultures, by the way, but they have some problems. It depends who you ask. I personally prefer to do DEC incubations or lab work, and they have problems with respiration and a lot of high bacterial densities. If you put a matrix in the water, within a few hours you get a lot of bacteria. So they are kind of hard to run. And then you have DEC incubation experiments, and these experiments have to be run for shorter periods of time, but they can be very informative. But one of the questions that I think we need to ask ourselves is, how long is a long-term experiment, or how long should you run your experiment for? And I'll give you a few examples of what we have seen in the lab and what other people have seen. So this is from Mueller, Antia, and Laroche, and they found, just if you look at this, so this is the cell diameter of Emiliania haxillii, of a strain of Emiliania haxillii under phosphate limitation. And this is the same strain under nitrate limitation. Yeah. And these are two conditions for N2P16-1. So this was actually quite disturbing when I looked at it, because all ecological models, which are mainly based on terrestrial ecosystem models conceptually, they use nutrients just as a black box. They do not distinguish between nitrate or nitrogen and phosphorus. But in this case, we see a response of different sign when you apply nitrate limitation or phosphate limitation. So this just highlights the fact that we have to, we can't ignore this. This is quite important. Of course, it, in a way, it screws up all, the way we look at these things, because we always thought that nitrate limitation could have an effect on, the cell volume and so on. But now it seems that, you have a different response if you limit the cells with phosphate or compared with nitrogen. So these are this is quite important. And also, if you look at the response, within days, if you run these experiments here, you probably wouldn't see much of the response. And sometimes you see a response, and then the organism goes back to the original condition. And we see that with cultures of dinoflagellates. That initially you see a response, and the cells produce more bioluminescence. I'm not going to show it here, and then they go back to its original state. So they kind of acclimate, and it takes them a few generations, and then they revert to the original state. So I guess what I'm trying to say is that we need to do some growth experiments, just monitoring what's going on with the cells, and see whether, basically try to design our experiments based on these observations, of the cells. And there are variables that one can look at, like FV over FM. We can look at the photosynthetic health of the cells. We can look at the shape of the cells, and the usual physiological parameters. But this is quite important to take into account. So so how many changes can be detected during this, if you like, acclimation phase or adaptation phase? So based on this, then you have to decide whether you want to go for continuous cultures or batch cultures or semi-continuous cultures, because different.
Batch experiments, the ones that are shown there.
Sorry, could you?
I'm sorry. Are these batch experiments?
Yes, these are batch experiments. Yeah. So I guess it takes a few generations. If I remember correctly, they use 16:1, 5:1, and 100:1, if I remember correctly, N-to-P ratios, and it takes quite a few generations for them to strip out one of the nutrients. So this is why it takes, and I can remember the growth rates. I don't know if they're here. So by day 11 or 10, by day 10, they reach the end of the exponential phase in the control in the N-to-P 16:1. So, but they observe this, by this time. So this is when they run out of one of the nutrients. So you have to decide also the variables that you want to measure. And then when you design your experiment, you have to make sure that you keep things as constant as you possibly can. I mean, there isn't such a thing as a perfect experiment, but you can just think about these things in advance. So for example, you have to try and keep the volumes constant, and you have to try and keep your head space constant. So if you remove your sample, you need to think about, supplementing this culture with precondition, pre-bubble, if you're working with in a bubbling system, seawater, and add up basically adjust the volumes. If you sample at time 0 in the middle of the experiment and at the end, if you sample more often, it's a lot of work, but you just have to do that. So we usually sample 3 or 4 times during the experiment, because there is nothing more that we can do. We have triplicates, and we have up to 4 conditions in the case of PCO2. So this is as much as we can do. But this is very important, because if you remove the volume, if you remove, for example, 1/3 of your volume, that will have an effect on the light irradiance that the cells are experiencing, and also you are altering the head space in your culture. So this is quite important. You need to account for changes in irradiance, and as a result of changes in cell density and in the volume of the culture. So as cells, as cultures grow to higher cell densities, you could have potentially light limitation. I mean, if you work with dilute cultures, that's usually not a problem, but you need to measure that and be aware of that. So the best thing you can do is to conduct trials to basically know the state of your cultures based on the cell densities, and you can calculate uptake rates and the threshold limits that your cells, can do well under and can not do so well under. And then cell quota calculations is quite useful. So if you know your cell quota, for example, if you run your experiment and you have your conditions, you have your cells exponentially growing, You can measure your particulate organic carbon, your particulate organic nitrogen, and that will give you your cellular quota. And then you can estimate at least the maximum cell density that you can have in your culture for your culture not to experience nutrient limitation, for example. And these are things that we don't tend to do unless we, think about it. So so then it's very useful to monitor nutrient changes during growth. And this is particularly important. For example, if you have a change in the life cycle within your culture, if you suddenly get formation of gametes, you might have a change in the nutrient utilization or in the carbon utilization in your media. So you need to know these things. And the best thing you can do is run these experiments in advance and get these numbers way in advance of your experiment. Okay, so this takes me about how much we know about the cell biology, so the inside, the guts of the organism, but also how much we know about the life cycle. So again, these are things that are really important. And as I said before, the case of EHACs, when they form gametes, they do not produce calcium carbonate, they produce organic scale, so this is biogeochemically relevant. And you also need to check under the microscope if you don't work with calcifiers, if you work with other non-calcifying algae. You also need to know that the original state and when you got the cells from the culture collection is the same as what you have now. And in all labs, we've all had problems with contamination, with parasites, with flagellates, or sometimes bacteria can affect your culture. So you need to keep an eye on all these things. And I'll just show you an example. This is from one of our cultures. We were actually very excited because we got gametes in this culture. So here you can see these are the coccoliths around the cells, and they stopped producing coccoliths. This is the organic scale, and we saw these flagellated forms. So we were very excited. We actually tested that these flagellated forms were Emiliani haxillii with molecular markers, and we were very excited because we realized that we thought that we could potentially induce gametogenesis in cultures. So then you have a really nice system to do lots of, in our case, a lot of interesting molecular work. We never knew how on earth we actually got these gametes, but this is important when you're growing your cultures, to know whether you're looking at this form or this form. And then we have sometimes in cultures cells that do not bear any leaves, any plates. Yeah? And sometimes in cultures, some strains are famous for losing the ability to calcify, like the strain 1516 in Emiliani haxillii, which is the one that has been used for the genome. This strain is extremely temperamental, so it loses it stops calcifying, particularly if you grow it in high nutrient conditions, and that's it. It stops calcifying, and it's very hard, and sometimes it's not possible to make it calcify again. So very important to look at the cells under the microscope. So this is just from the paper by Trimborn. So these guys, what they did was to alter the calcium levels, and they looked at calcification. And I'm just showing this to highlight or to illustrate the different forms that you can be looking at within the same organism. Of course, calcium is semi-conservative in seawater, but I'm just showing you different calcifying forms, generated by different environmental conditions. So the question you need to ask yourself is, which one is representative of the field, or which condition are you actually looking at, and how you can compare that with the field. There are media that have 3 times more bicarbonate than natural seawater. There is a media recipe that everybody or many people are using that has the wrong magnesium to calcium ratios, and not only the ratios, but the calcium levels are very low, so the cells sometimes show malformations of coccoliths. So it's very important to pay attention to the media recipes. Yeah. So if you want, I can give you some examples a coffee time, but it's really, we were using a recipe for a while, a few years ago, and I had a look at some of the cells, and I checked the recipe from that, published work, and a recipe a lot of people are using, and the calcium levels were 1/3 of what they should be. So they were about 3 millimolar. So if you grow your cells for a while, they could experience calcium limitation. So this is very important. And of course, if you look at just natural populations and representatives of populations, we have a naked strain, a non-calcifying strain isolated from here, from the equatorial Pacific, and then we have these two strains. And I always show this because this strain produces 10 times more calcite than this strain here. Yeah. So this is the problem that we have within what we call the species concept. If you're a modeler, this is a nightmare, because which one do you use of these two? So we need to be aware of this, and not only this is pretty constant in culture in terms of calcification rates, but there are other strains that, for example, this 1516 that just changes calcification if the nutrient availability changes, and so you need to be aware of that. So and these are the original populations where these guys were isolated from. So I was trained. As a biologist, you are trained to do your experiments in the lab, and then you field test what you found in the lab. But I tend to think the other way around now. So I like to go to the field, make my observations in the field, and then I try to come to the lab and work under tightly controlled conditions to test whatever I observed in the field. But this is quite disturbing, because if you use the concept of species to define your model, is not very useful, really. And then you have another complication in the field, that is giving genetic variability between and within populations, and these are viruses. And I'm just showing this to illustrate the fact that they are really important, and the fact that they are huge. So this is a virus, and the size is within the size range of bacteria. And these viruses are quite interesting, and we thought that they were species specific, but they are actually strain specific, which is incredible. And this is just for just as a piece of anecdotal information. So these guys produce these compounds called ceramide that are used in cosmetics, and they infect the populations. So they release these compounds. They are thought to prolong the life of blooms, and when the cells cannot cope any longer with the number of viruses, they burst, they lyse, and then you get the programmed cell death kicking in, and all this release of organic matter and so on. But let me show you this slide when Kay Beidel from Rutgers University and colleagues. So they were working on these two strains of Emmeliania huxleyi, so the strain 374 and the strain 373. Yep. So and they infected them with the so-called Emmeliania huxleyi specific virus. And what they found is that the strain 373 was resistant to this virus. Yeah, so we have a lot of information for these viruses. This virus is actually sequenced. Its genome is sequenced, as well as the Emmeliania huxleyi genome. Now, one could question how useful this information is if you have this not species specificity, but strain specificity. So yeah, there is a lot of complexity. And here you can see the abundance of cells, and here you can see the FV over FM of the cells. So you can see that the 374, which is this strain here, drops, after just a few hours of infection. The photosynthetic efficiency of the cells is, basically lysed after a few hours. So this is really interesting, from a if you are interested in genetic diversity and species diversity in general. And I also wanted to show you another example, and this is from my lab. So I have a program on bioluminescence, and we are working with these bioluminescent dinoflagellates that produce scintillons, which are these organelles that harbor luciferin and luciferase. So when you change the the pH of the medium, or when you, introduce any physical disturbance, when you shake the cells, you get the production of light. So luciferin turns into oxyluciferin, and there is light production accompanying the reaction. So the green spots that you can see here, I think is the blue is chlorophyll and the green is the luciferin, and this is what we can see with confocal microscopy. And what we thought is that we would see this circadian control of scintillon production and of luciferin synthesis. And what we find is huge variability within cultures that have been synchronized to the light-dark cycle, that have been grown for years under the same conditions. So we have, in this case, I'm showing here 14% of cells that do not contain scintillons at all, and then we have 20% of them that have more than 10 scintillons, and the literature values report about 300 scintillons per cell. Yeah. So what we know is that within these cells we find a lot of variance. Within cultures of Emmeliania haxillii, we find different degrees of calcification. We have cells that have several layers of coccoliths and cells that have one coccolith. So there is some variability that, again, we need to take into account. And so we tend to assume that our cultures are synchronized because we grow them constantly in this 12-hour light-dark cycle. We give them the same nutrients. We keep them exponentially growing, or whatever. But we need to actually check that this is the case. But there is always inherent variability within and between independent experiments. Yeah. So so we always find variability between our replicates. So the more replicates you run, I guess that's the answer to finding the mean in the response to your experiment. So when you report on morphological traits, it's very useful to report on some quantitative measurement. So it's good to show images, for example, of SEM, but it's good to provide a value, of percentage of, brittle leaves if you work with coccolithophores, or in larvae, if you look at spicules, not just showing an image, but show the percentage of larvae in the culture in your incubation that have that characteristic. So if you read the literature, I think more often than not there is no quantitative information associated with SEM illustrations, for example, of to scanning electron micrographs, and I think that is quite important. So the way populations change, we don't know in terms of what takes over, or whether if you have a clonal culture, whether at some stage for a particular for particular nutrient levels or light levels or whatever within the evolution of this culture, whether there is a switch on of a particular function that will give you a different physiotype, if you like. And in the natural populations, we just don't know how populations change. This is probably unlikely, but still, we don't know whether we have a kind of like a multi-bloom event of dominant genotypes or ecotypes, if you like, that take off and bloom at different stages of population development with a background of, the genotypic pool of the population. But this is probably quite useful to keep in mind, when you are thinking about what does your strain tell you about the natural population. So this is what I was referring to before, that we have, these bloom-forming species here in the high latitudes, and we know that we have different genotypes or ecotypes, within species, and we are focused on these bloom-forming strains that we collected during the blooms. But actually, the biggest contribution to export production doesn't come from these very striking blooms in the high latitudes, but it comes from the lower latitudes. So again, something to think about. So how representative are our clones of the natural populations? And this is another issue that one needs to take into account. That we work with clones that have been kept in culture collections for a long time, and again, we don't know about the selection bias for those particular clones that are well adapted to grow in the lab, and we don't know about the effect of these clones having grown in artificial conditions in the lab, how they might have changed their, their physiology and their morphology and so on. So this is quite important. So in terms of the long-term incubation experiments, what happens in the oceans with respect to what happens in a bottle where you're growing your phytoplankton. So you have several million organisms in a liter of water during bloom conditions probably. Then you have several generations in a year. So if you think about how many generations does a population experience over a year, well, I don't know, but certainly, when we grow the organisms in the lab and we expose them to these abrupt conditions, there is a mismatching between the change in the rate of, let's say, increase in PCO2 and the number of generations the population undergoes. So the question is, when organisms evolve, how do we find them? How do we see that change? How do we see evolution? So we have a number of clonal isolates that we got. This arrow should be the other way around. We got from the field, and again, how well do they describe the natural conditions, and how can we run these experiments, to have a realistic representation of the field? So we have these monocultures, these clonal cultures, and then we have mid-scale incubations, and then some people work on larger scales, and they introduce pulses of mainly nutrients, but we tend to work with anywhere between here and here. And again, one could think about having these reservoirs and using these reservoirs to get samples for as much as you possibly can. So we tend to grow large volumes. This would be a huge cell density, so that we don't work with these kinds of cultures. But we work with diluted cultures, large volumes, and we harvest material for as many parameters as we possibly can. And one of the harvesting that we do is for proteomics. So we try to harvest protein from our vessels and look at what changes under different conditions in different PCO2 conditions. Okay, don't know why this is not moving. Oops. Okay. And in these manipulations, I wanted to show you this graph because this is something that came up a few times, and when we discuss omega and when we discuss the manipulation that we use in our experiment and so on. So we have these two. If we are thinking about calcification, we have these two potential sources of carbon for calcification, which go in different directions when we increase the PCO2 in the media. Yeah. So we need to think about. Essentially, I don't think we know for most organisms whether they use this form of carbon or this form of carbon for calcification, and this is central to the question of how organisms acclimate or adapt to environmental selection pressure. So this is I think really central to really central to a lot of the questions we are working on. And also, we have to think about the way we represent, we investigate the changes in processes like calcification. And more often than not, you see them expressed or plotted against CO2, when CO2 is really physiologically not a driver for calcification in any way. Of course, within the size, within the ranges that we are looking at. So I think this is, in my view, extremely important. So this is one, one aspect, and another aspect of this is the implication that this has got. So that this has biogeochemically. So this is a coccolithophore cell. So you have here the chloroplast and the coccolith vesicle, and there is lots of things bringing ions in and out, and some intracellular CO2 concentrating mechanisms and so on. But essentially, if the cells utilize bicarbonate to form calcium carbonate, there is acidification of the medium, and there is discussion about whether there is coupling between calcification and photosynthesis and so on. However, if the cells use carbonate ions, there is no acidification of the media. So this is interesting from a physiological point of view, from an evolutionary point of view. These organisms that use carbonate will likely be affected by, the way we are moving to our lower carbonate seawater. But the organisms with more sophisticated pumps, like in the case of bicarbonate users, they probably require more sophisticated proton pumps or calcium ATPases, and we know that there are calcium ATPases pushing calcium out of the vesicle as well as into the vesicle. So these are very interesting unknowns that we are trying to work with. Now this is really difficult because the conversion rates between bicarbonate and carbonate, as you know, are of the order of, as I understand, 2 seconds. So.
Microsecond.
Oh really? So I don't know how one could do this, and if anyone has an idea, that would be fantastic. But I think this is a really important question. But I also wanted to point out that I was talking to John Raven about this, and the pH of intracellular compartments is quite low. So these are, when we think about thermodynamics, we always think about external to whatever cell or organism we are working with. And although organisms also follow the laws of thermodynamics, these reactions are not in equilibrium. So we need to think about what's going on inside the cell. So not easy, but I think it's quite important. And I know people have been talking about this over the last couple of days, so I just wanted to mention that it has been reported a potential mechanical effect of bubbling by Xi, Morel, and a third author, I can't remember his or her name, but so you have to pay attention to these things. The bubbling that we do is extremely gentle. We work with very high PCO2 levels in our gas cylinder compared to what we have in the bottle. So we just introduce very high PCO2, so we end up with approximately half. So we, in the case of when we want to achieve 750 ppm VCO2, we are bubbling 1500 ppm VCO2. So that gives you an idea. We just have an open system and bubble extremely gently to achieve the desired, the desired ppm. So when you run an experiment, decide on the number of replicates that you want to have, and at least you should have 3. So we tend to have more. This is for proteomics, but we tend to have 4 replicates. And just think about your cell quotas and the maximum cell density you can live with in your culture for whatever conditions you want to be at. So in our case, we try to keep them at around, below 50,000 cells per mil. In our case, we grow just under 15-liter cultures. And think about the fact that you need to look at your physiological data in conjunction with the chemistry. So you need to think about your blanks and your controls, and when you have to sample. So you need to sample at time 0, and you need to sample before you inoculate the cells, and you need to keep your control without the cells and sample there as well to see how that has changed. I don't know how I'm doing with time. Okay, how long do I have? Until. Okay. Okay, so just to emphasize that bubbling must be gentle, or you must bubble elsewhere and then use that preconditioned seawater in your culture. And cells have to be checked for physiological stress. So remember that some cells can form cysts. Remember that some cells can, be disrupted by an abrupt change in the condition, or by high bubbling, by robust, vigorous bubbling. Just ask yourself whether you know your organism's physiology, and try not to make assumptions. If you don't know, just think about what extra manipulation you need to do, what extra condition you need to have in your experimental design to account for that unknown. And this will save you time, because more, it's quite likely that you might have to do it later anyway. Another thing is that I just wanted to bring this up, and I will talk about this a little bit tomorrow. But when you look at, we are looking at formation of, well, we are looking at net calcification, but we don't know what is being formed and what is being dissolved. And one of the things that's particularly for, for very high PCO2 levels is that we need to think about whether, about the mineralogy of the organism we are working with. And magnesium, the magnesium contribution is important in assessing the susceptibility to dissolution of the biomineral. Yeah, so for example, if your organism forms low magnesium calcite, which is percentage of magnesium calcite below 4%, then you're on the safe side. But if the organism produces, I think it's above 8%, if I remember correctly, of high magnesium calcite, I believe the susceptibility to dissolution is comparable or even higher than that of aragonite. Yeah, so these are really important considerations, and we don't know these percentages in coccolithophores, for example, and in many calcifiers. So there is some information for some echinoderms, and Mario Lebrato, who's working with Andreas Oschlis in Kiel, is doing some work on that with us.
So is it a problem with magnesium substitutes in?
Yes, yeah, and the thing, because the susceptibility of dissolution increases, the higher the magnesium content is in the calcite.
Below 4, it's not significantly different.
No, but there is data for echinoderms showing pretty high levels of magnesium contribution, and also in coccolithophores is probably not the case, but in animals, in many animals, there are changes in the biomineral from amorphous calcite to low magnesium to high magnesium calcite, and sometimes you have half of the animal producing calcite and half of the animal producing aragonite. So it's yeah.
But just magnesium calcite less than 4% is essentially calcite.
Yes.
In terms of
Yeah.
It's more like 12% when it comes.
Yeah, and I think in echinoderms, if I remember Mario's report, I think he found up to almost 20% magnesium carbonate, which is really high. But this was, for a group of echinoderms, and it's probably not representative of everything, and it's probably not a problem in coccolithophores. But I don't think we have. It's another consideration, perhaps. Okay. And this is wrong. These are not unknowns. We know that calcification generates protons using bicarbonate, but it does not generate protons using bicarbonate. So these are things that the organisms that see acidification within the calcium carbonate vesicle or reservoir, they require these proton pumps and these calcium ATPases to keep things at the right pH. And this is something else that I wanted to show. So as well as nutrient quotas, as well as in relation to, as well as the threshold for tolerance of the different organisms, we have the specific K not 5 CO2 for photosynthesis. And in this case, I'm showing Emiliania haxillii and let's say skeleton imacoste. So a diatom, a coccolithophore, and this is Phyocystis. Yeah. So you can see clearly here that in the case of the diatom, you can see here the CO2 requirements for the cells to achieve the maximum levels of photosynthesis compared to in the case of Emiliania haxillii, which have a particularly high requirement for CO2. So they have low K half CO2. So this is, we assume this is again, species or group specific, but this was done in strains of these organisms, and we know that if you try different strains with different properties, this K not 5 CO2 can change significantly. So something else. So as I was saying before, will you be able to compare your data with previous results? So can you justify using the same conditions? If you can, go for it. If the previous protocols can be improved, then change the protocol, but it's quite useful to be able to compare your data with what has been published before, and also of course report your data, submit your data to data repositories, so then it can be used by the wider community and to get the bigger picture. Okay, so I just briefly wanted to talk about the meta approaches and how useful they can be, to look at the bigger picture from a cellular basis, to look at the mechanistics of the different processes and their high and low CO2. So, Craig Venter and Ed DeLong and Mitch Sogin, they started this meta-genomics approach. So they just looked at populations and pulling the mixed cell communities and looking at the diversity of these communities by looking at genes. And then what we are trying to do is use proteomics. So we have a paper in proteomics in review, undergoing the second review, which essentially describes the proteome of several strains of Emmeliania haxillii. And what we are doing now is analyzing the data for an experiment, for 4 experiments, for replicate independent experiments that we did under current CO2 and under 750 ppm VCO2. So we did these experiments over the last few months, and we collected all the data that we traditionally collect, as well as harvested material to analyze the proteome. And these are, for those of you who are not familiar with these kinds of techniques, we have the genome, and this is based on genes and in non-coding regions as well. And these are static, if you like, descriptions of organisms. But we can go further, and we can look at the functional level of organization in the organism and look at the proteins that are indicators of, they can be indicators of function, and they can give you an idea of how the system is evolving, how the system is adapting to change. Yeah. So, what we hope is to be able to develop this, to optimize or to apply this method to the field. But this is optimized for lab work, so we can distinguish between strains that have different physiological properties. And this is something that you don't necessarily do with genomics, not necessarily do with genomics. So this is another level of information that is telling you about what the cells are doing rather than who is there in the water. So when you do proteomics, you need to, when you do any of this kind of work, you need to check with people from other disciplines. So it's really important I talk to chemists a lot, but I also talk to geologists because sometimes you get a lot of information just by giving them a sample or just asking them, what can we give you, or what conditions are good for you, and that kind of thing. And then something that we try to do is, based on these kinds of conversation, we have a backup plan. So we collect extra samples for different things. One example is to collect, for example, samples for SEM or samples in formalin or whatever, or glutaraldehyde. You can always analyze them later. So even if you are not thinking of doing any morphological work, it's a good idea to collect these samples, and if you have any problems with interpretation, you can always go back to your samples. Another thing that I wanted to talk about is the time of sampling. I talked before about replication, but the time of sampling is another consideration. And we tend to do our measurements during the light phase, because this is when we go in the chamber and the lights are on, because it's easier for us to work when the lights are on. But I'm thinking about whether we need to look at both the light and the dark phases. And I've been thinking recently about the oxygen to CO2 ratios and how much we know about them. In these kinds of experiments, we never measure oxygen, which is quite easy to measure. And I, as a physiologist by training, if you look at the activity indirectly, the activity of Rubisco, for example, I think it's really important to look at both, to look at these ratios and how these these activities or these rates change with a change in the ratio, especially if you are looking at peak-to-poke ratios and things like that. So this is an example of the experiment that I was telling you about that we did over the last few months, and we are now dealing with the samples. So this is the kind of planning that you need to do. So we work in these really large vessels, and we maintain these cultures for generations. So this gives us about 10 generations of evolution, and we take these kinds of this kind of sampling, so we monitor what's going on, when we are pre-acclimating the cells, when the cells are undergoing acclimation during the experiment, and then we can go back and see if something went wrong during the experiment. And then, of course, at the end, because in this case we are limited by, we need a lot of cell biomass. We need to harvest for eye track for proteomic analysis at the end of the experiment. We can only harvest once, but at least we know that the sample, the cells have been pre-acclimated for quite a while. And because you will get information about the subcellular location of the proteins that, let's say, are upregulated or downregulated, then you can look at biological processes and what is being upregulated or down. So here I have an example of membrane-associated proteins and nuclear proteins and so on. So this is for the static, proteomic proteome paper that we did in exponentially growing conditions with modified F/2 medium. But what we are doing now is looking at two conditions. So instead of different cell lines exponentially growing, we look at the same cell line under two conditions. So what we hope is that we will find some interesting protein expression associated with one condition or the other. So the way we prepare the samples is really important to obtain relevant results. And it's important for the applications that you are going to use later on. And you need to assess, the parameters. Some of them you can only assess them qualitatively, but try to work on quantitative parameters like up and down regulation of genes, and we can do this with eye track, with quantitative proteomics, and also work with bioinformaticians for these statistical considerations. And one example about the sampling, the kind of sampling that you need to do, well, you have this protein synthesis that happens in very short time scale, so you have to work as fast as you can, basically, and then we can try to use this protein synthesis inhibitors like geneticin or cycloheximide. But these protocols don't work for all cells, so this is something that's you can do with some species but not with others. And if you cannot interrupt protein synthesis by using these compounds, for example, you have to think about how you can work really fast to have a realistic representation of what went on at the time of sampling, rather than these weird heat shock proteins that you are getting because of it took you a long time to centrifuge to spin down the cells. So what we typically do is snap freeze after the manipulation for protein extraction, and then so this is really important to have very reliable storage facilities. And depending on what you're looking at, so for DNA is easy, for RNA is not easy, and for protein is probably a nightmare. So you have to think about how you need to store your samples. And then you need to, if you're looking at gene expression or protein expression, you have to think about these kinds of things. So you can have a gene that is present in a clone, but it could be not expressed. It may not be expressed. And then there is a lot of work on transcriptomics, but there is not necessarily a coupling between the transcriptomic level and the proteomic level. So there is this halfway between DNA and protein that we are looking at, but I think it's quite useful to look at both levels. So I'm just going to give you an example of these guys from Lenski's group in Michigan State University. So they worked with Escherichia coli, and they achieved 40,000 generations of evolution. So they've been growing them in a specific medium, culture medium, and they have been subculturing them daily since 1988. So this is a hell of a lot of work, and you need a lot of hands to do this. But basically, this is what they found, that bacteria become more ecologically fit, if you like, within the first 2,000 generations. Then the cells become bigger, and most of the gain in the organism comes from 5 different genes that have mutated. So that's what makes them bigger. And after 20,000 generations, they sequenced this number of bases from 50 isolates, and they found 10 changes in all organisms with a mutator genotype. So they identified 1 mutation over 20,000 generations. So this is the kind of thing that I think, we need to think about, and we should be moving towards. So so these are the evolutionary considerations that I think we need to bear in mind. So can we assess evolutionary adaptation with our current protocols? Probably not. How old is your strain? So your strain has been adapting to culture conditions. So that's probably an interesting evolution experiment already. That if you have the time 0 for that strain, and then 20 years later you're using that strain, well, how representative is that? How comparable is that with how it used to be at time 0? Yeah. So, and if you put this in the time, I always like showing this quite dense slide, but this just shows the huge, time and spatial, variability, scaling problem. So in terms of the molecular processes, you have reactions happening in milliseconds, or in seconds, or in minutes, and then if you are in the field, if you want to relate your lab work with the field work, you have all this added complexity of transport and mixing and so on. So I just wanted to leave that with you, and this is key for looking at how organisms change their physiology, how new physiological traits they acquire, what physiological traits they lose in the process, and also the who wins. So in the adaptation of these organisms to pressure, which one takes over, and if they take over, how are they behaving physiologically? So I think these are the questions we are all trying to answer. And I'm just leaving this slide there for you to look at the kinds of things that we I think should be looking at. So we are looking in the lab, and we are looking at biology or functionality, using diversity to look at all these things, and we take physiological measurements. Now we have the luxury of looking at large-scale patterns, and we have all these nice technologies, from molecular technology to large-scale monitoring. So I'll leave it like this. The last slide is acknowledgements, but I'm going to leave it there because hopefully there will be some questions about this. Thank you. Yep.
How does working with non-acidic cultures confound your interpretation of proteomics results?
Well, with proteomics, you only pull out the most dominant proteins. So if you keep the bacteria levels really low, the chances of getting a dominant protein from biomass, which is orders of magnitude lower than the actual eukaryotic culture, is very unlikely.
So you monitor bacterial cell counts in your cultures?
Yeah, and through the culture, at the end of the culture, when we harvest. Also, we can identify bacterial proteins quite easily. So we don't see that as a problem. And some people use antibiotics, but I prefer to stay away from antibiotics. But as I said, if you keep your cell densities really low, work with large volumes, you should be okay. In terms of the subset of proteins that you pull out from Emiliania haxillii in this case, we only see the most dominant protein. So if a protein is produced in in low abundance, we won't see that. So you don't pull out all the proteins, just the most dominant proteins. So we get, I think, something like 150 major proteins. Yeah.
If you want to look at the evolutionary change of this, and then what is your recommendation to manipulate the carbonate system? To me, if you want to look for many generations, shouldn't you just gradually change the CO2 system instead of, suddenly have 750?
Yeah, this is what we are proposing to do. So the idea is to have incremental pulses of CO2. And of course, the calculation of the generations, the number of generations that a population undergoes every year is very hard to calculate, But we make the assumption based on, cell densities at the peak of the bloom and the background numbers. So we think that with 1,000 generations, we might be looking at definitely at decadal timescales, but possibly even at 1 century.
Deborah, in one of your slides, I think you showed that if cells are using bicarbonate, you said they would evolve a proton, but if they were using carbonate ions, they wouldn't. And I think that, the chemist can correct me if I'm wrong, but I think that's incorrect, that if there's calcification, there's CO2 evolved and protons produced.
But if this, we haven't tested this because we don't actually know the source of carbon for calcification. But if the cells produce CO2, so if they are acidifying the medium, they need to exert a better control of that intra-reservoir, intravesical pH, right? So my guess is that they will need calcium ATPases and proton pumps working more efficiently if there is a change in the pH given by calcification, right? This is, I don't know, we haven't tested this, but if they use carbonate, I don't think there is acidification of the media, as I understand,
yeah,
right?
There definitely is. If you just think of black clots, ultimately they're using carbonate. Maybe they brought a bicarbonate atom across the cell membrane, but ultimately it's a carbonate that's reacting with the calcium and then the proton.
Right. Oh, that's something I learned. So ultimately, it's the carbonate ions that they use in calcification, Chris. So is that what I just want to make sure that? So ultimately, it's the carbonate ions that they use in calcification.
So there are carbonates that you're actually bringing into the internal compartment. They are producing synthase within that compartment if they lock in carbonate. Or if they lock in bicarbonate, whether it be an internal proton generated. The effect in the media is the same, but in the internal calcium compartment, you could have different responses, whether you take a carbonate as the specific source for calcification versus bicarbonate versus CO2.
I don't think so. Andrew, you care?
Well, I think that he helped embellish more clearly there, because you're right, the medium won't change. You've either got a bicarbonate breaking up to a proton and carbonate which goes into the cell, or bicarbonate goes into the cell and the proton gets thrown back out. But internally, I think it's also going to matter what other counterions are brought in with this and how they're moved around in the system. And the simplistic view of simply saying it has to be a proton in this part of the cell is not yet proven.
Mm-hmm.
Yeah, Michael is right. What happened is that the bicarbonate releases transforms into a carbonate outside the vesicle, and then you won't have a proton generated inside the vesicle. But I don't think that they are transporters for carbonate ions. I'm not sure it has been shown before. They are transporters for bicarbonate, but I don't think that it exists for carbonate ions. So I think eventually it is the bicarbonate which goes inside. And not
this is what we think that's the case. I think it's very hard to, we don't have that information for carbonate, but I wouldn't, discard that as a possibility. And for me, it's one of the most interesting questions, because if they bring bicarbonate into the vesicle, they certainly would need a mechanism to control, to buffer the pH inside the vesicle. So the question is where in the cell there is. And in the cytosol, there is this conversion of CO2 into bicarbonate to control the cytosolic pH. So, I mean, if they calcify, they certainly, there will be acidification of the media somewhere, either in the cytosol or in the vesicle. And the question is whether, if it is within the vesicle, then they would certainly have to have a sophisticated way of buffering the pH. But yeah, we don't know.
Just wanted to comment. I mean, I know nothing about the physiology, but if I was designing a system where I wanted to precipitate calcium carbonate, I would have to have it in a container where I could pump protons in and out to manage this. Mm-hmm. And so your buffering concept, I mean, it's not really a buffer at all. It's an active system looking to run this. But I don't think you're going to easily distinguish between the examples you're given. I mean, at a certain level, you might say, yes, these channels have been shown to be appropriate to these ions. There are undoubtedly many channels we haven't yet seen. So, the question is, how much do you need to know to get on to where you are there? The classic example we had recently in a paper Dave Checkley and I had, where for fish otoliths, it was clearly obvious that what seemed to be happening is that there was physiological control of pH in the compartment, but it couldn't keep out CO2 gas. And so carbonate ion was going up when PCO2 went up.
Right. Yeah, I mean, one thing that we have been doing is looking at the activity of calcium ATPases and the expression of calcium ATPases, and we are looking at 4 types. And 3 of them bring calcium into the coccolith vesicle, but one of them extrudes calcium. So then you can look at calcium uptake, but to design these experiments, to look at whether they are bringing in one or the other form of carbonate or bicarbonate, I don't know how to do them. If anyone knows, I would love to.
So, if I understood you correctly, it matters how you try and lower the pH, isn't it? If you're using...
Everybody agrees with that, I think, yeah.
Have you tried this yourself and found out what would be ideal for non-calcium carbonate from organisms?
The method for lowering the pH for non-calcifying, I wouldn't make any distinction. I think in my view, I mean, I prefer to use bubbling to have my gas phase for which I know the conditions and bubble that in the media. We are doing that with dinoflagellates now, and with some dinoflagellate species, it can be a problem because if you disturb the media, they form cysts, so you don't want that. But what we do is we measure the FV over FM with different bubbling, and because we use very gentle bubbling, we have been able to do that with the species that we have, but that's not always possible. And in that case, I would then bubble a separate reservoir and do daily subculturing, which is a lot of work. But that's that's my choice. There are sometimes you cannot use the same protocol because of logistical impossibilities, because your reservoir is too large. And one thing that I would avoid is using these ceramic tiles with lots of pores, because they are really disruptive to a lot of phytoplankton species I've worked with. So if I use that, I lose the culture within just a few generations. So I would stay away from that. But this is my personal opinion. I think the organisms don't care about alkalinity, but they care about the sources of carbon. So I think if you adjust alkalinity, it's probably okay. I mean, I always think about, I always try to keep the system, use as much as I can natural seawater, instead of artificial seawater. And if I can bubble, I would avoid manipulating the system in any other way. But sometimes it is appropriate to use, to adjust the alkalinity and manipulate it by adding acid or base.
For us non-biologists, I picked up on culture medium you were talking about, and you said that one of the recipes that had calcium, for example, at 3 millimolar. How standardized are the culturing medias across labs or within labs?
Mm-hmm.
And if these cell lines have been grown for multiple generations at 3 millimolar,
Yes.
how long does it then take for them to come back up to conditions or be acclimated to growing back at 10 millimolar calcium?
Yeah. Well, when we get them from the culture collection, it depends on the culture collection. Some use organic media, some use inorganic media. Most of them use really high inorganic nutrient concentration. So, as I said before, the F/2, which is typically used in, by many scientists and in some culture collections, uses 880 micromolar nitrate, which is really high. I mean, we use 880, almost 900 micromolar nitrate. So that's crazy, I mean, it's really, really high. But and we still use very high, nutrient levels. So we make the, if I want to compare my data with previous work, I need to, compromise on these things, and also sometimes you need to enhance growth. So you need to, push up your nutrients a little bit. So we tend to use 100 micromolar nitrate and 6.24 micromolar phosphate, so 16:1 normal ratio, and we work with that, and we keep the cells exponentially growing but really low cell densities. In terms of adaptation, what I can tell you is that when we put them under our conditions, they seem to acclimate well. We get really high growth rates with the lower nutrient concentrations in our medium. For example, the NCDH, which is a high calcifying strain, the one I was showing here on the right, that grows, with in these conditions at least 1 generation per day. So it's, it grows really well. Usually it's just above 1 generation per day. So, and in F/2, I don't know, it probably grows even faster. But yeah, I mean, the adaptation to nutrients, I think, is quite, for the fast-growing strains, is not a problem. The problem is when you have a strain which is picky, which is temperamental, and more often than not, we just don't know what makes them, grow well. And it could be metals, or it could be vitamins in the medium. I don't know. We supply the media with vitamins. So, I mean, Craig Venter's team has this really neat way of isolating, single cells. So they have this impedance approach using flow cytometry, and they picked droplets with one single particle in them, and they just place them in these, matrices where they can grow. It's really amazing. So they have their own media, if you like, and they can maintain them and look at. They can do they cannot do single cell genomics, but they can use these cells to assess diversity. So we cannot do that, unfortunately. But yes, I don't know about the evolutionary trends in 20-year cultures.
So I guess there are a number of species that we'd like to study that are not easily cultured.
Yeah.
Are there techniques being developed? Is there any hope of culturing pteropods in the future, or is it you got what you got?
I'm probably the wrong person to ask. I've never worked with pteropods, but what I hear is that you can only keep them in the lab for nothing, for just a few days. I mean, it's a similar, we have a similar problem with larvae of echinoderms. If you are doing an incubation experiment and you spawn the larvae and then you look at, fertilization, and you follow them, the larval development up to metamorphosis, for example, I mean, you have to ask yourself, is this okay? When you see high mortality, you probably have to stop your experiment. I don't know. I don't have a clue about how to grow pteropods in culture.
one comment and two questions. To answer Christine's question about theropods, I have a student working on theropods. He's able to keep them for 5 to 6 weeks
Wow.
in the lab. But eventually they die, probably because they need very large volumes of water, like cubic meters. And obviously that is a problem for perturbation experiments because we can't afford to put one theropod in 3 cubic meters of water. So that's for theropods. We do keep the larvae very well. We collect the eggs and we bring them to juveniles. That's no problem. The problem is when they become adult and want to move up and down in water column to feed. That's where the problem is. And that's when they require very large volumes. With respect to the cultures, you mentioned the batch cultures and the fact that the culture media have a very high isothermic concentration. Don't you think that the use of hemostats would enable to perform experiments at a more realistic level? Like that means that you could do that under
Absolutely.
nitrogen limiting.
Yep. Yeah. No. Definitely. And especially to look at this long-term adaptation, I don't think there is any other way to go, other than continue, chemostats, and just keep the this range, this really narrow range for a long time. And in the case of ocean acidification, with incremental pulses of PCO2, every whatever number of generations. This is what we are planning to do. So of course you need an army of people who are taking care of different aspects, of the chemostat. So it's not trivial, but I think it's the only way to go.
One more, if I can. You mentioned Lenski's work and the fact that he has E. coli for in culture for 20 years, and I believe that some of the EH strains have been in culture also for a very long time, perhaps as many years. Do we know in which carbonate conditions they are kept during those years?
No.
We don't.
I don't think so.
pH levels, no?
Yeah, my guess is that because they used to be maintained at really high cell densities, not anymore now, but sometimes they had to re-isolate, from these cultures because they got contamination and stuff like that. But no, the carbonate chemistry, I don't know. They use natural seawater. For example, in the Plymouth collection, I think they filter sterilize the seawater, and of course there could be some, subtle probably changes in the carbonate chemistry. I don't think there are any measurements on the carbonate chemistry, unfortunately.
But some labs have moved to cryopreservation.
Sorry?
Cryopreservation of the cells.
Yes, but Jean-Pierre was referring to the actual carbonate chemistry. So, I guess the reason why you're asking is because if the cells were isolated, these clones were isolated, I don't know, we have clones which go 40 years back, the carbonate chemistry then could be, quite different from the carbonate chemistry now from, waters from off Plymouth, for example, when they, so that would be really, really cool to do, but if we had the information, but I don't think we do.
I think we'll delay any remaining questions for the break. We'll reconvene here at 10:30. I think Deborah should put